So I'm going to start with sequencing technologies. It all started with these sequencing technologies. So in 1977, Sanger method. So you use one of the first methods for sequencing. In 1981, human mitochondrial genome sequence was obtained. And in 1990, Human Genome Project started. And the founder of the project was my thesis advisor, Professor Dr. So we are the first bioinformaticians working on this project. In 1995, complete cell genome was obtained. 96, complete eukaryotic genome was obtained. 2001, supposedly, Human Genome Project was obtained. But back then, they could sequence fragments that are around 1,500 long DNA sequences. So they had to cut the DNA into small pieces. They got several copies of the same genome and cut it from different locations so that they get overlapping fragments. After sequencing, they knew the short fragment sequences, but they didn't know the order information. You need to find the order of these fragments to be able to combine them and get the whole genome sequence as a result. So they ended up with millions of fragments, but they didn't know where it belonged in the genome. Then they came to computer scientists. They said, okay, I have these fragment information. How can I combine them to obtain the whole genome? And that's where bioinformatics started. So that's the first application of bioinformatics. They turned this problem into a graph problem. They took every fragment as a node in the graph. And if there's an overlap between the end of the sequence and the beginning of the other sequence, that means they can come after each other. So that gives the order information. So by combining these graphs and finding out how much they overlap the fragments, they established the graph. And the links between them were directed links. End of this is linked to the beginning of this fragment. And the weight on the link was amount of overlap between the fragments. And then they established this whole graph. And they use Hamiltonian path. Hamiltonian path is a path that connects all the nodes with maximum weight. So that makes sure that you're combining all the fragments all together with maximum overlap between the fragments. Because two fragments can overlap with one letter by pure chance. But if they are overlapping with 20 letters, you're sure that they're coming after each other. So you know the order information. And this is called Hamiltonian path problem. And there are algorithms in computer science to solve this. So they applied these algorithms to this problem to get the human genome sequence. So that's the first thing. The complete human genome, it wasn't complete in 2001. Actually, it was completed recently, 3 years ago, because there are parts of the genome were not covered. Especially the Y chromosome was very difficult to sequence. So it was not obtained back then. But the working draft was obtained in 2001. It cost billions of dollars for the Human Genome Project. Today, you can get your sequence done in 2 weeks for $1,000. Soon it's going to be going down to $100 range. So everyone will have their genome. Every species will be sequenced. We will have their genome information. And in 2005, we got the next generation sequencing for 54 sequencing technologies. And for this part, actually, the second generation is mostly the eliminal, the dominant one right now. But that can sequence short sequences. So you have to cut your fragments into 150 long on average lengths. And then you can accurately sequence them using the second generation sequencing. And using the second generation sequencing, they conducted the Human Microbiome project. There are lots of information available there. If you're working on this type of research, please use them. And then third generation sequencing, long read sequencing technologies started in 2011, and Nanopore, Oxford Nanopore came up with even Nanopore sequencing. It's very small impact, so you can carry out the whole sequencing instead of these big machines like PacBio and stuff. You can do the sequencing everywhere, so that enables sequencing even in space. It's very portable. You can take it everywhere. You can take it to any environment. You can take it with your boat to do aquamarine sequencing and carry out the sequencing over there. And now the third step is using these long-read sequencing. We have the Human Microbiome Project. Third stage of the Human Microbiome Project started in 2019. So shotgun sequencing are the first ones. So breaking the DNA into small fragments. And without the reference to genome, you can do the assembly like we did in the Human Genome Project. You can get fragments and then establish these graphs and combine them to obtain the whole genome sequence of a novel genome, de novo genome assembly. So you can do whole genome sequencing. So instead of taking, you can find the structural variants and lots of information about coding regions and non-coding regions at the same time. And that was done in around 2016. Whole exome sequencing focuses on only the exome regions. And you can extract these regions using specific primers. Extract these regions and just sequence the exome regions, because exomes are supposedly the most informative parts. You have the gene information, and then mutations in this gene will be reflected into the protein sequence, so you can find the protein sequence information as a result. But there are issues with this one. Certain exomes are not obtained. It's skipping certain parts of it because of this primer design issue. So you have to know which method that you use to extract the exome and whether there are uncovered regions, especially for human applications in West. We had several cases that the doctor was sure that the patient has certain disease, and they sent the sample to Centogene to sequence, and Centogene came up with the report saying that it doesn't have that disease because that person does not have any mutation in relation to that disease in any gene, but the clinician was sure that the patient had that disease. Then we looked at their exome sequencing, and in a very important gene, it wasn't covering the first exome of the gene because of the primers that they were using. It wasn't picking up that region. It wasn't sequenced, and there was a mutation. We did the Sanger sequencing of that region, and there was a mutation that's causing the disease that the geneticist suspected. So if you do West sequencing, you have to make sure which part of the exome is covered, whether there are mutations in the parts that are not covered that can be related to your disease if you are applying this to diagnostics or your species as well. But with microorganisms and so on, it's much better to do the metagenome sequencing, the whole genome sequencing. Then we started with targeted sequencing approaches. If you have certain region that you're targeting, it's much cheaper. You're sequencing a smaller length. And 16S ITS, 18S rRNA regions. 16S rRNA is used for bacterial identification. And ITS targets the fungal species. 18S RNA is used for eukaryotic microbial analysis. So you can do all of these targeted sequencing analysis. Then the next was RNA sequencing. So today, using the NGS technology, we can sequence the RNA. And actually, we can even do more than this. We can do single cell transcriptomics. You can measure the RNA at single cell level. And now we are doing spatial transcriptomics. So you can find the location of the cell that you are sequencing, and you can combine them with the images as well of the tumor tissue. For example, there is a difference in the coloring in the tumor tissue, and you sequence the cell from that region and from the neighboring region, and you can identify differences, especially in terms of tumor. It's heterogeneous. There is the clonality of the tumor, so there might be different clones available within that tumor that can correlate with the image and the transcription signature. And you can do direct RNA sequencing without reverse transcription, ddNA conversion, and methylation sequencing. Recently, that's the big thing. You can sequence the whole methylation pattern without doing even the conversion. Using these long-read sequencing technologies, you don't need to do bisulfite conversion. You can sequence it from the signal that you are getting. You can find out which parts are methylated, which parts are not methylated. This is the new application of the sequencing technologies. As I said, first generation Sanger, second generation Illumina biosequencing, short reads, PCR-based, high throughput. Third generation is nanopore and PacBio. So what do you do? The workflow is you have to get the nucleic acid extraction. For that, you can use chemical lysis or mechanical lysis, physical disruption using beating or pH shift to get the DNA out of the organism that you're trying to sequence. So mechanical lysis is much better in that sense because it might be difficult with chemical lysis in certain type of species that you're trying to sequence. Then you have the library preparation step, which is platform specific. So you do fragmentation and barcoding PCR in Illumina, and DNA is shared, and adapters with unique barcodes are added via PCR to each fragment, so you know which sample it's coming from. That barcode is telling us. In nanopore, we are using barcoding PCR. It gives platform-specific barcoding of fragmented DNA. You can also do native sequencing, though, with enzymatic ligation. So in this case, that's with on nanopore as well. You don't have PCR. You have adapter ligation using ligase enzymes directly to native DNA without the PCR step, because PCR can create certain mutations as well. So you want to eliminate these type of risks in sequencing. And then you run the sequencing in a chosen platform. So comparison of these technologies, short reads 100 to 300 base pair, but it's highly accurate, especially the Illumina is extremely accurate. But you don't get structural resolution from the short reads. If there are big structural changes, you cannot obtain it from short read technology. And it's fragmented, so you have to do fragment assembly. So you have to run a specific algorithm. And best use cases for short read technologies are variant calling and polishing, like finding out final detail of short sequences. But the major frame should be obtained by the long read sequences, especially if you are dealing with the novel species. So long read sequencing, you can get more than 10,000 base pair. What's the longest we got? Berkay, what was the longest DNA sequence?
800K.
800,000 long. So if you can get very long DNA, if you optimize your DNA extraction process, if you can get it without breaking it, you can get the whole piece, 800,000 long piece sequence altogether without cutting it into smaller pieces. But compared to short read, it's not as accurate. It creates some errors, read errors itself. Now it's much better actually with the advancements in this technology. we are getting close to the accuracy of Illumina. It has excellent structural information. You can find out missing pieces, inversions, deletions, and so on using the nanopore technology because it's capturing the whole area. So you can find out whether there are missing parts, inversions, and so on using this nanopore technology. And since it is very long, it's very easy to assemble these pieces. You don't need to bring these small pieces because small pieces, you can have alternative solutions. So you can misplace that fragment to a different location, and that can change the sequence as a result. And you can obtain novel species and complex genomes using this long read technologies. And usually, if you want to get very high accuracy whole genome sequence of the novel species, you do hybrid sequencing. Nanopore gives you the overall structure. You can obtain the nanopore sequencing and then map the short reads into the scaffold that's given by the nanopore technology. And then that gives you highly accurate because Illumina short reads don't have much error. So you can get accurate with the whole new genome using the hybrid technology. So if you're going to identify anything novel, I don't know what kind of sequencing service you're taking.
Whole genome.
The whole genome.
Yeah.
Okay, using nanopore or Illumina?
Illumina.
It's not sufficient. You have to do a combination.
Ah.
Yes, it has to be hybrid. Okay. So, okay, yes, of course. So, if you are going to get a novel species, you have to do it by hybrid. If you have money only for one, do the nanopore instead of Illumina. For novel species, if you don't have a reference genome, Illumina doesn't work because it tries to assemble the fragments, and there are repetitions. It can put the small fragment equally likely to different locations, and then you get the wrong sequence as a result.
But after 16 RNA sequencing.
You're doing 16S sequence.
Yeah.
Even in that case, we will discuss it's not very useful. Okay. So, applications. The best platform for 16S is nanopore. Lots of short read tools, but has misclassification at genus level. Illumina denoising and a true SNP confusion. So whether it's a denoising or whether it's a SNP. And there are a very limited number of short read databases. So that's why nanopore is much better, especially for certain species like Shigella and E. coli. When you do the 16S sequencing, especially the regions that the Illumina is sequencing, they are identical to each other. And Shigella is a pathogen. You have to be treated for it. E. coli, no harm. So if you do the Illumina sequencing, you don't know whether that sequence is coming from Shigella or E. coli because they're identical in the region that you are sequencing. So you don't know whether there is a pathogen or a harmless bacteria in your sample. Even with long region, there are very few regions where they differ. Especially bacterial sequences are very similar to each other. They have 7 or 8 differences, and the rest is identical. So if you have a small fragment coming from the identical part, you don't know where it belongs to. So you get the wrong abundances as a result using the short read sequence. Only the parts that's covering where the variations occur, that can give you whether that fragment is coming from E. coli or Shigella. But if it's coming from the conserved region, you cannot identify where it belongs, and you get the wrong abundances. Species level information, for sure ONT, because ONT covers the whole region. So you know where it comes from, that sequence is coming from. Decreased cost and less time consuming. Rapid field sequencing, if you're in the marine in a boat, you want to sequence the samples. ONT, even in space, they are using ONT. Structural variant determination, ONT. Clinical workflows, Illumina, because it's approved by the FDA. You have a reference genome, human genome applications, for example. You have a reference genome, you can map the reads to the reference genomes. In that case, you can use Illumina because it's very, and it can determine the mutations. Then de novo assembly, you have to use both hybrid sequencing. What's happening? Okay. Why is microbiome diversity important and how it is measured? It's a marker of ecosystem health. Diversity is the sign of health. Even in our gut, higher the number of species that we have, healthier the gut is. And if you feed on specific type of food all the time, even though it's healthy, it can dominate your microbiome, and it doesn't allow space for other good bacteria to grow. So you have, for example, if you're consuming too much kefir and yogurt and cheese or fermented goods, they have high in lactobacillus strains. Even though it's beneficial, you have to have a certain amount of lactobacillus. If you're over consuming this type of food, they will dominate your gut, and in such cases, we are seeing that another good bacteria called Bifidobacterium disappears because it doesn't allow any space for Bifido to grow. So that's why you have to have a balance and you have to have variety in the gut in any healthy environment because they all carry out certain function and we need their function. So if that bacteria is not growing because of overabundance of another supposedly good bacteria, then you have lack of that function in your gut, and it's going to cause a disease as a result. So that's why it's very important. Low diversity has been linked to diseases such as inflammatory bowel disease, obesity, metabolic syndromes. High diversity may promote functional redundancy, improving system resilience. Alpha diversity, that's within sample diversity, and it can be measured by metrics like Shannon and Simpson indices. You're going to learn how to calculate these things. So within the sample, how diverse the species are. And then beta diversity is between the samples. So it quantifies the dissimilarity between microbial communities in this case. And richness is total number of distinct taxa present in your sample. So metagenomic sequencing, including 16S rRNA gene amplicon sequencing or shotgun metagenomics, allows you to have the microbial taxa and genes coming from those species at organism levels in a sample. And from the gene information, you can go to pathway information, which pathways are triggered, which metabolites are produced, in how much abundance, and so on. You can pass the composition information to the gene information and gene information to the function information. So what's functional annotation? That's what we are saying here, that which metabolic pathways are triggered as a result of the abundances of these bacteria. So each bacterial strain has different set of genes expressed, and they trigger different metabolic pathways as a result. So short chain fatty acid productions and so on are triggered as a result of these. So, and using this, functional annotation is the process of predicting biological function of genes identified in metagenomic data. It links DNA sequences to proteins, enzymes, or pathways, they encode, helping to understand what microbes are doing, how they interact with the host, and what are they triggering within the host, and which pathways they are activating or deactivating as a result of the metabolites they are producing or the genes that are being produced, and so on. And why is it important? It helps us to reveal the metabolic potential of microbial communities, sulfur cycles, nitrogen, carbon cycles, It allows us to detect genes related to antibiotic resistance or virulence. It offers insights into host microbe and microbe-microbe interactions as well. How is functional annotation performed? We have gene prediction. So metagenomic sequences are scanned for open reading frames, and you can identify novel genes. There are gene prediction algorithms. You can identify a novel gene and you can annotate the function of that gene, depending on the similarity of that gene to other organisms with a known function. Database matching, you can predict that genes are compared to databases like KEGG, EGNOG, PFAM, CARD, and other databases. And then you can find out, you can do the functional annotation, like biosynthesis of ether secondary metabolites, glycan biosynthesis and metabolism, and so on. There are several levels of information you can obtain by functional annotation enrichment. Okay, so far I'm introducing the sequencing technologies and how they are applied, and Ali will talk about the relevance of microbiome studies in aquaculture health. Thank you.
Hello, everyone, and welcome. Before I start, I would like to mention that I have been a bit sick lately. So please excuse me in advance for any short coughing during my talk. By the way, my name is Arslan, and I'm currently working with Prof.Dr. Uğur Sezerman in Acıbadem at Acıbadem University. I'm also the manager of epigenetics and biotechnology company focusing on genomics and microbiome-based research. I would like to express my sincere gratitude to Dr. Semih Alpsoy for inviting us for today. In this presentation, I will talk about relevance of microbiome studies in aquaculture, health and productivity. But a little bit is VGS, maybe whole genome. First one, we generally use the sequence technique for disease detection. Detect early to prevent outbreaks, go beyond traditional methods, use long read sequence of eDNA. The eDNA means environmental DNA. Identify pathogen at strain level for ONT technology and PacBio technology, not Illumina. Enable on-site real-time diagnosis. Recent studies show that nanopore sequence enables portable on-site pathogen genotyping. Zarantello nanopore enable microbiome analysis in rainbow truth aquaculture, and the Delamere rapid genotyping in Tilapia lake virus using nanopore sequence. These methods mark the beginning of real-time molecular surveillance in aquaculture. This is the nanopore-enabled microbiome analysis in rainbow truth aquaculture, figure 1. Description for presentation shows the sampling to sequence workflow in rainbow truth aquaculture. The different type sampling, A, B, C, D, E, A, water, B, sediment, C, gill mucus, D, skin mucus, E, gut content, and gut mucosa. There is a workflow. Sampling is sampling to sorry about that. Sampling to each sample undergoes nucleic acid extraction. Minine sequence, bioinformatic analysis, detailed microbiome profiling. This one is Delamere work in Journal of Fish Disease. The figure shows the complete workflow in rapid field-based genotyping of Tilapia lake virus using nanopore sequence. This study demonstrates how portable sequence device can be applied directly on fish farm to detect and identify RNA virus within a few hours. Almost 12 hours is done. First step is sample collection on farm, RNA extraction, Tilapia lake virus PCR, gel electrophoresis and controlling. Standard amplicons, the 5 steps. 6 is nanopore multiplex library preparation and minine sequence and mapping to reference genome. At the end of the pipeline, read to map to reference Tilapia lake virus on genome, and consensus sequence are uploaded to NCBI to public data share. This slide is genomic data is more than analysis, it's tool for management and decision making. Feed optimization and water quality monitoring. The second one is Bruno 2023 BMC microbiology. Feed and water microbiome analysis in rainbow truth ecosystems. So in this first slide, I will walk you through the overall scope of the study. The researchers aim to investigate how an alternative feed made from Tenebrio monitor or mealworms affects on just the gut microbiota of rainbow fruit, but also the microbial communities in the tank water. Yes. What makes the study comprehensive is the broad sampling strategy. They didn't just focus on the fish gut. They also sampled from the tank water, biofilm, fish skin, intestinal mucosa, and even the feed itself. In terms of methodology, they used to 16S RNA gene sequence and whole genome sequence. The combination gave them a more complete picture, both taxonomically and functionality. They also performed a detailed network analysis to study how microbes move between different sources, like from feed and water or water to fish. The novel species discovery ensemble. The figure shows workflow of the bacterial genome reconstruction and novel species identification using both short and long read sequence technology. The process began with bacterial isolate followed the DNA extraction and sequence on two platforms, Illuminasec short reads and nanopore minine long reads. The Illumina data are assembled using space, the nanopore data with fly or unicycler, and both are combined in hybrid assemble approach. After assembly, the genomes are compared to quality MLST typing and core genome phylogeneity. At the bottom, you can see the analysis virulent genes and mobile gene genetics elements, which helps determine not only taxonomy but also pathogenic potential of newly identified strains. Altogether, this approach allows researchers to reconstruct the complete bacterial genomes and reveal previously unknown species or strain. Caption of slide the hybrid genome assembly workflow for identification of novel bacterial species using illuminated nanopore sequence. The sequence technologies are a core driver aquaculture digital transformation. By combining short, long, and hybrid read approach, we can move from the data action. Multomic integration, combining transcriptomics, metagenomics, and metabolomics, enable environmental and physiologic decision support systems. Sequence technologies no longer read just the genome, they read the ecosystem. Thank you for all. My slide is done now.
Actually, I would like to thank Ali for these applications using sequencing technologies. We have one paper in Microbiome that was published last year. I want you to read that paper. It was from the space station. They collected samples from the space station and sequenced them. And those were the bacteria that was on that space station when it was in Earth. And when it went up to the space, due to the conditions, infrared and the change in the gravity and all these things, it changed. The sequences changed. So they sequenced all the samples and we did the analysis in comparison to the Earth species, how much it diversified, and what were the genes that were highly mutated as a result of the adaptation to space conditions. And we looked at the gene level information. There were certain genes that did not change, maybe because the changes in those genes made those species extinct. And there were certain genes that had accumulated lots of mutations, and those genes were metalloprotein genes. Accumulated lots of mutations, and mutations were proline to glycine mutations, or a big hydrophobic residue to glycine mutation. As you all know, glycine is highly flexible. Prolines are very rigid. So this mutation made the protein more flexible, adapt to the conditions in the space. So that's the publication that we had in Microbiome last year. So, and that enabled the metalloproteins to function for the biofilm formation and for the species to survive this harsh conditions in the space. So it's a very good application, actually. When you obtain an old species, how you can annotate the genes, how you can find the mutations, and how can you regulate it to the function and the survival of those species. So you may have similar things in this harsh Arctic environment. What are the adaptations? Which genes are mutated in comparison with its counter like in other places? And you can find out adaptation changes compared to Arctic conditions, for example, high infrared as well, and also the temperature is another constraint. So check, it's Microbiome. It's published in Microbiome. Put my name, Sezerman Microbiome Space Station. You will get that.
What about human? Did we just proceed with the bacterial population and did we find out for human as well?
From the space, we have some microbiome changes as well.
In human?
In human, yes. Actually, that I wasn't on the paper, but my student Cem Meydan was on the paper. They had twin astronauts. They kept one in Texas and sent the second one to the space. They had exactly the same diet, and they looked at the microbiome at the beginning, and the twin lived in space for close to 3 months, and they compared the microbiome changes, and it changed a lot. Actually, the twin was much younger than the one in Texas. Under space conditions, the epigenetic markers, they looked at the epigenetic markers related to aging and so on. The twin that lived in space was much younger than the Earth condition, and microbiome changed completely. Even though they had the same food, microbiome changed completely.
So also looking at the gene expression,
Yes,
that also
yes,
changed.
yes, a lot, actually. So even the phenotype changed a lot. The guy was younger, thinner. They had exactly the same food. Nothing changed, actually. So there are big differences that you can do such comparisons. And for that, they use nanopore.
But
So using immuno, especially V3, V4 region, they are sequencing all day, not sufficient to give you detailed information at species level.
I want to talk with you.
Yes, you can talk to Ali as well. That's the whole situation. Yes.
yeah, I was asking, what could be the environmental causes for them? Because they try to keep the environment in the space station as close as possible, except for the gravitation.
They have to. The organism living in space condition, they have to adapt actually.
I mean, gravitational effects is the major thing.
Yes.
But other than
And
that,
infrared and all these light
they are
exposure.
protected for radiation.
Yes. There was like closest species to the one that we reported. There was 65% mutation rate. So it mutated a lot to be able to adapt to the condition. And there were some novel species as well that were identified. So it's an interesting study. And you'll get, there are many publications coming out of the space station sequencing. And if you're doing aquamarine, there are from our group lots of sequencing from aquamarine. Chris Mason is we have a consortium, Metasub, and we are sequencing every environment in the world. So underwater, deep water, volcanoes, and all these extreme conditions. So, and we are identifying the genes, annotating them, what's happening to live under this extreme condition, and so on. So read those papers.
Interesting to understand, what are the limits of adaptation.
Yes, yes,
It's like, for instance, the tardigrades, right? They can adapt, so much, they can stay in space environment for years and come back, still adapt.
yes, certainly. Actually, it's like the selection process. I'm sure there are certain ones that are dying, but the ones that are surviving are the ones that can adapt to that harsh conditions, actually. So it's a selection process. Okay, thank you. This is the first part of our talk.
I have a question.
Yes.
So most of the papers you read about aquaculture use biosensor data, environmental data, such as pH, temperature, and they want to use the machine learning or deep learning approaches. So how could metagenomics or other omics data could contribute to?
You will learn. That's the second part of our talk. Okay.
Then, second question. We are doing multi-omic approaches to combining data. So there are various data that are not multi-omics. How can we just integrate all of this data?
That you will learn as well. You're going to have metadata and omics data integration approaches. You will learn. That's the topic of today's meeting.
Excellent.
Okay. Thank you.